Hi everyone, my name is Nur Atika Liana. I want to discuss about gene expression, which is it involves two processes. The first process is transcription and the second process is translation. What is gene expression? It is the process by which a gene's information is converted into the structures and functions of a cell by a process of producing a biological functional molecule of either protein or RNA is made. The process of gene expression is used by all known life, which is eukaryotes, including multicellular organisms, and prokaryotes, bacteria, and archaea. Gene expression is assumed to be controlled at various points in the sequence leading to protein synthesis. RNA plays an active role in all stages of gene expression. There are three types of RNA that involve in protein synthesis. First, messenger RNA. Second, ribosomal RNA. And third is transfer RNA. Messenger RNA. Messenger RNA is produced in the nucleus by a process called transcription. Messenger RNA carries genetic information copied from DNA in the form of a series of 3 base code words, and each of which specifies a particular amino acid to the cytosol. Ribosomal RNA associates with a set of proteins to form ribosomes. The two ribosomal RNA subunits join with protein in the cytosol to form the subunits of ribosomes. Ribosome attached to the endoplasmic reticulum to synthesize protein. Transfer RNA is a carrier molecule for amino acids, delivering them to the site of protein synthesis. As there are 20 different amino acids, there are also 20 different types of tRNA which bind it and carries it into the growing end of a polypeptide chain. Overview of gene expression. First, both coding and non-coding regions of DNA are transcribed into mRNA. Introns removed during initial mRNA process. Exons splice together and the spliced mRNA molecule is out of the nucleus through addition of end cape and a poly A tail, the mRNA used to construct protein. Pathway of gene expression. Firstly, the DNA is transcribed to mRNA in the nucleus and it is called transcription process. Next, mRNA is translated into protein in the cytoplasm and it is called translation process. Pathway of gene expression in detail. First, DNA in nucleus serves as a template for mRNA. Second, mRNA is processed before leaving the nucleus. Third, mRNA moves into cytoplasm and becomes associated with ribosomes. The fourth step is tRNA with anticodons carry amino acids to mRNA. The fifth step is anticodon-codon complementary base pairing. And lastly, polypeptide synthesis takes place one amino acid at a time. Protein synthesis is the process in which cells build proteins from information in a DNA gene in a two major steps. First, transcription, and the second one is translation. Protein synthesis requires the use of DNA and all of three forms of RNA to read the code and make the protein. There are two major steps of gene expression, which is first, transcription. It is synthesis of an RNA that is complementary to one of the strands of DNA. Second, translations. Ribosome read and messenger RNA and make protein according to its instruction. Next, let us proceed with the first major step of gene expression, which is transcription. It is a synthesis of an RNA, which is mRNA, that is complementary to one of the strands of DNA. It takes place in the nucleus. Transcription produces an RNA sequence complementary to one of the DNA strands, which is part of the DNA, serves as a template for mRNA formation. Uses an RNA polymerase enzyme which binds to a region of the DNA. Pathway of transcription. First, RNA polymerase attach to DNA at a sequence called promoter. Second, DNA strand separates temporally. Third, RNA polymerase adds RNA nucleotides to the DNA template. Fourth, RNA polymerase travels along template strands. Fifth, RNA polymerase continues to read from 3' to 5' until it reaches a point on the DNA strand called a termination signal. Lastly, DNA helix strands joins back together. Next, the second major step of gene expression, which is translation. It is a ribosomes read and messenger RNA, which is mRNA, and make a protein according to its instruction. It takes place in the cytoplasm. Sequence of mRNA bases complementary to those in the template DNA. It determines the sequence of amino acids in a polypeptide. tRNA assists by bringing amino acid to the ribosome. Step in translation. mRNA binds with a ribosome at the start codon. tRNA brings amino acid for the polypeptide. Second, ribosome reads the codons and assembles the amino acid in order. Lastly, stop codon release polypeptide and ribosome.